Our last step in discussing inheritance is a discussion of gene linkage. And gene linkage answers the question, what happens when two genes are not on different chromosomes? Remember, if like gene A and gene D are on two different chromosomes, and you have an individual that is doubly heterozygous, there are 4 unique gamete types that are produced. However, if gene A is really close to another gene on the same chromosome, independent assortment no longer applies. There aren't homologous chromosomes carrying these 2 different genes to separate in meiosis. So for example, meiosis 1 would separate these 2 homologous chromosomes from each other. Big A would always go into a gamete with big B, little a would always go into a gamete with little b, unless there was some kind of crossover event between these two during meiosis 1. So we're going to look at the consequences of segregation and inheritance when genes are linked or close together on the same chromosome. So imagine a purple flowered plant with long pollen, purple being dominant, long pollen being dominant. And this plant is doubly heterozygous. Now when we self-fertilize the plant, we would expect to see a 9 to 3 to 3 to 1 ratio of purple long to purple round, which is the recessive version of pollen, to red, which is the recessive version of color, long, to red round, 9 to 3 to 3 to 1 ratio. Instead, if these are not found on the same chromosome, we would expect a 9 to 3 to 3 to 1 ratio. And instead, we see a different observed number of phenotypes. The reason for this is that the genes are linked. So, for example, I see more purple long and more red round than I see of either of these 2 classes, the combination of the 2. That indicates to me then that these 2 alleles are linked on the same chromosome, and these 2 alleles are linked on the same chromosome. And in meiosis 1, if there's no crossing over, gametes have either the 2 dominant alleles or the 2 recessive alleles. Now, if you look at the Punnett square, these are the combinations that we get, and we only get purple long and red round. What we haven't accounted for is the presence of these 2 classes of offspring. Those will come about by crossing over. A crossover event in prophase 1 will combine big P with little l and little p with big L. Now it only involves 2 chromatids rather than the 4, so there will also be 2 gametes produced with 2 dominant alleles and 2 recessive alleles. However, this crossover event is rare. Crossing over is less common than no crossing over, and so these gametes will appear in greater numbers than these gametes. These will appear at roughly the same amount, but they will definitely be in larger numbers than the recombinant types. So I have a gray-bodied, long-winged fly, doubly heterozygous, mated with a black-bodied vestigial-winged fly, which is homozygous recessive. From this mating, I would expect to see a 1 to 1 to 1 to 1 ratio of all of these different combinations of phenotypes. However, instead, I see 2 larger classes, gray and long, black vestigial, and 2 smaller classes, gray vestigial and black long. These smaller classes must be recombinant types, or recombinant types that come about by crossing over in this heterozygous female. We ignore the male because the male is only homozygous recessive, and so even with crossing over, he can only give little g and little l. The larger classes represent those without crossing over with the 2 dominant alleles, and those without crossing over with the 2 recessive alleles. The number of recombinants over the total number of offspring equals the percent recombination, or the recombination frequency. That can be directly related then, directly applied to a distance between the 2 genes on the chromosome. the distance between these two genes on the chromosome is 17% recombination, or 17 map units, where every 1% recombination equals 1 map unit of distance. To explain it again, the male can only give this kind of gamete. Females give either the parental type gametes or the recombinant type. Offspring, then, are a result of the combining of these types of gametes. It's actually nice to use recombination frequencies in order to come up with distances between genes along the same chromosome, and also the order of genes along the length of the same chromosome. Imagine 3 genes, and one of them encodes body color, one encodes eye color, one encodes wing length. To figure out the distance between them, you can figure out the recombination frequency between these 2 genes, between these 2 genes, and then also between these 2 genes. 3 different experiments where you mate a doubly heterozygous individual with one that's homozygous recessive. Between G and C, there was 9% crossing over. Between C and L, 9.5. Between G and L, 17. Now, of course, these don't add up to 17. They add up to 18.5, but that's because every experiment is an independent event. We would say that the distance is about 9 map units between these two and 9 between these two, with this one in the center. So recombination frequencies and analysis of linkage helps to determine maps and map distances of genes along the length of chromosomes.